website. We also just sent all of them to you on Slack about 25 minutes ago. Basically, our panel of judges up here will rate each team on these 4 criteria with a score of 0 to 3, giving everybody a score of 12 points per team. I believe there are 8 judges, so approximately a score of 0 to 100. We will count up the scores after, and then we will announce the winners at the happy hour. Does that make sense to everybody?
Yep.
Any questions on that? Scoring just became 1 to 5. So, you know, it's about 160 points total.
A little bit, but that's yeah.
All right, so let's get started with some introductory remarks. So Ono, who is also doing hackathon stuff, is also sort of running the podium for the presentation. So big round of applause for Ono. I think we'd like to speak to pretty much all of you during the hackathon. And if you haven't spoken to Ono yet, you should catch him during the happy hour. I mean, I'm sure he would be very excited to speak to you. Speaking to Ono is a lovely thing. Yeah, I think the other people to really thank are Pete, who found me and really sort of logistically put this event together, and Laura, who is really the only organized person you see in the front of this room. That's really it's been really nice watching her work, and she's really held everything together. And then I really had fun with all the judges, and actually I didn't know really any of these people before, but it was amazing to me. So Carlos was a little late, and he got stuck in traffic coming the first day. And the first thing he said to me was, "I'm going to get some water. Would you like some water too?" And I just thought that was such a nice way to meet somebody. Like, you know, so that was kind of cool. And in these hackathons in general, I mean, I think you know, when I ran my first one, I told people it changed my life, and I think everyone changes my life a little bit, and this has been really special meeting all of you. I really mean that, and I hope it's been special for you too and changed your life just a little bit. And hopefully it changes the life of some people a lot. And we hope it changes science a little bit too. And I've seen some really cool scientific projects out there. And you guys really have been amazing because you come from non-homogeneous backgrounds, right? Like a lot of the hackathons I run, people are from very homogeneous computational biology backgrounds. You guys are all over the map. There are people that are professional computational biologists. There are people that walked in here and work on whatever you work on and learned enough about genomics to find something cool. There's one project that I think is particularly amazing in that sense. It's been an amazing experience. And Ono, did you want to say anything before we start?
Yeah, like, since I get the opportunity, I don't know, it's been a really surreal weekend for me. Like, I was sharing this earlier, like, 3 very different days. I can say, like, Friday when we started, like, a lot of excitement. There was really excitement and so many people showed up to work on this. You know, sharing my data, working on a real problem. I saw all the brain power in this room. It's quite impressive. So that was Friday. Then Saturday, I felt there was like a general high here. Like everybody was so like into it and working like crazy. And so my eyes were completely wide open the whole day, and I have to say it was hard to fall asleep after that day. It took me a while before the tiredness finally took over. And then today I feel, I sense a lot, everything got more realistic, right? Like people come up with real things, they identify genes that might be affected in me, or how to move forward. And you know, the talks, the discussions, everything is like way more real and down to earth, which is kind of a good transition because I feel, oh my God, I wish this was over because I have to get down to work. There's so much to do. Yeah, so I want to thank everybody for showing up and yeah, together like, create this amazing like weekend that I will not forget. Thank you.
That was amazing. So now we are going to move to the presentations. The first presentation is by a remote team called Team Edward. Awesome. So Team Edward, I was hoping they would send me 3 to 5. Are you guys here?
We're Team Edward.
I mean, since you guys are up here, why don't you go ahead and present? All right, you have 4 minutes. Go.
All right then. Right then. Good afternoon, everybody. We are Team Edward. My name is Kevin. This is Mustafa, Josh, and Hide. And our project is SNP and indel density cluster ranking.
So we wanted to see if NF2 is driven by genes outside of NF2, and if the inactivation of this gene is what causes phenomas and other diseases.
So our goal was to locate the NF2 region and then measure the SNP and indel density in the NF2 gene, and then find a pattern in the density. Then we use this to compare the genes without NF2 to see if they have similar characteristics to the pattern.
Okay, so we retrieve the data from the ClinVar database and UCSC's HG19 gene data. We retrieved the SNP and indel data using bed tools from the VCF files. And for the testing data, we took Anno's tumor mutation data and subtracted Anno's brother's non-mutation data with Ensemble. Thank you, Anno. Thank you, brother. Okay, so our methodology was to find the SNP and indel density in the testing and training data. And then we used a k-means clustering algorithm to characterize a gene by its distribution of SNP densities, SNP and indel densities. Okay, so given these density features, we created a cluster model, and we used the cluster model to identify genes with similar features.
So as for our, I think next slide. So as for our current progress and the kind of results that we've come out with so far, so we're able to actually successfully train this k-means clustering algorithm to group these sequences that we're studying by the SNP and indel densities. And because we just started running out of time, we only attempted to study a small set of our entire test data. But we were able to calculate the SNP and indel densities for our test data and for our training data, but we just haven't been able to yet rank the test data that we were getting out. Yeah, we just didn't have enough compute time. We were literally just maybe half an hour out from finishing. But our biggest result was that we were able to successfully train this cluster model to recognize NF2 SNP density patterns. So to conclude, next. Sorry. Oh, no worries. So to conclude, our original goal was to find other non-NF2 genes that cause the NF2 tumor to develop. We took existing NF2 variant data from ClinVar to train a clustering model to identify genes that have similar SNP characteristics. And we attempted to use this model to understand non-NF2 genes. So we could use this tool in the future to identify SNP and indel characteristics to annotate genomes for studying other diseases or for like understanding and studying genes that may cause these NF2 tumors. So in the future, oh yeah, next. Yeah, almost there. In the future, we want to use other metrics other than SNP and indel density to cluster data together. And maybe this might look like studying the SNP prevalence in the 1,000 Genomes Project or the allele frequency. We also want to study a larger data set. I know we were limited by time today. And we wanted to create a better fitting cluster model by varying our cluster parameters. And eventually we might want to explore integrating this with some kind of neural model. But yeah, we really want to thank you for listening to our presentation and for the wonderful opportunity of being here today. Yeah, thank
you. We have time for one question. Don't move. Are there any questions for this team?
I wanted to be Team Jacob.
And you guys didn't finish the results, but any indications of any genes that have similar SNP and indel propensities?
So that's something we were just like right at the cusp of. Oh yeah. We were pretty much right on the cusp of figuring that out. Maybe, I don't know. Maybe if.
Did you see any like 3-4 letter gene names that popped up that look kind of similar? No one's gonna hold you to it.
Yeah, not yet. We're still getting results. So yeah.
Actually, there were some trends that we found. We computed similar.
Team Jacob for the win.
Yeah. It's actually, my name's not actually Jacob. So.
All right. I would like to say about this team, they quietly worked. They were here late. They worked like crazy. Big round of applause for this team. They were awesome. Thank you. All right. You guys can exit stage left. So, Ono and I have decided to switch up the presentation order. So we're going to bring team Deep Drugs up now. And then after that, Ono will pick teams at random to present, which I think is a lot more fun.
Deep Drugs?
Deep Drugs. So I believe this was a team that spoke to me the least. So maybe that's why they were so successful. I don't know. They also seem to have more than 6 people, but you know.
rotating lineup.
Oh.
Not everyone was here all the time, but collectively we're here now.
Gotcha. So each of them was 86% of a team member, roughly. Hello, we are Deep Drugs. Okay. Who's presenting? Okay.
It's going to mirror the display. Get a really small Deep Drugs.
No, it's fine.
All right. Hi, everyone. Yeah, like you said, we are Team Deep Drugs. My name is Ryan.
No, we don't.
Okay. Well, and this is my beautiful team. And we're here to talk to you today about what we spent our 3 days doing, and that was exploring novel and off-label therapeutics for NF2. So I'm a pharmaceutical engineer by training, so I was happy to bring into a project that I thought would leverage my biological background while also being able to pull on all the amazing AI and machine learning colleagues that I have around me. And so I don't think this issue really needs to be addressed. We know that NF2 does not have a currently existed targeted and effective therapeutic. And so our goal was to try and explore biologically mechanisms that we thought were of interest and that we could exploit towards developing future therapeutics for this disease. And so currently there's this large research database that's been growing towards the identification of druggable targets for NF2, but I think there's a lacking paradigm right now between the disease space and current existing therapeutics that really understand what these pathways are leading towards and what's actually causing this cancer phenotype to appear. And so our first goal was to really dig into the literature and find out what exactly is happening and what we think might be suitable drug targets. And so just based off these pictures, this is very simplified diagrams of some of the relevant pathways that we think are occurring in NF2. And from a biological nerd standpoint, NF2 is a really cool sort of mutation because it's a weird state of cancer where it not only affects extracellular receptors, but it also is interplaying with canonical pathways that are occurring inside the cell. And so you have this interplay between receptors that are feedback looping on the outside and then interplay from within the cell. And so when we were looking towards druggable targets, two things really stuck out to us. One of the canonical pathways from the extracellular receptors was an interplay with the inner receptors, and also there were finally targets that were not just general cancer targets that we often exploit, as you can see, like the mTOR pathway right next to the red box of targets that we're interested in. And so looking through this list of targets, we began a literature search looking for what we thought were interesting small molecule proteins that we thought could exploit some of these pathways towards inhibiting the development of NF2 into cancerous tumors. And so towards that, we utilized DeepChem and the macromolecular docking to then take each of our proteins of interest and then evaluate their binding affinity in relation to the ligands that we had selected. So going back to this pathway, looking at RAC1, CDC42 dual complex, the PACs, the JNKs, and RAS complexes. So leveraging DeepChem and its macromolecular docking platform to then evaluate the suitability and affinity of those pairs.
That's fine. Like, as part of getting DeepChem to work, we submitted 5 PRs and had them merged. So thanks, Broth, for merging them. So we were able to use the DeepChem web app to input various pairs of ligands and proteins and find their affinity scores. So.
We were encouraged when he told us not to use the GUI to, in fact, use the GUI. And so here's a sample run of us doing just that. Oh, there's no sound. I will say we spend an uncomfortable amount of time adding music to this video, so the fact that there's no sound is actually quite upsetting. And then yeah, so that last point is a readout on affinity scores for those pairs. And so we just want to highlight what we found was the most interesting. So this is CDC42, one of the markers that I was talking about, and then the drug target, the drug itself, AZA1, is a current explorative therapeutic. It was our favorite going in just based off its biological mechanism, and it ended up being the strongest binding affinity in our macromolecular docking studies. So that was a really positive result. Maybe some bias involved, maybe not. And this is just a proposed mechanism for what we think might be occurring when we look at what AZA1 is doing. So we think that through impediment of the RAC1 CDC42 complex, it impedes PAC1, and then pushes these tumor cells towards the cell survival, cell death mechanism, which forces them into apoptosis. So normally these cancer cells, they continue dividing, they continue progressing through the cell cycle without actually going into apoptosis. We're hoping that this mechanism recapitulates that and might show not only specificity for NF2, because we know that RAC CDC42 is not present in all forms of cancer, but also may have implications in other forms of neurological cancer. And yeah, future work. Obviously, this was a very small subset of hand-curated molecules that we went through with a strong biological basis. I love the work that's going on with natural learning and language to be able to parse through thousands of articles and be able to pull targets. So doing something similar and then plugging into DeepChem would be great. Also, of course, I'm from the lab, so wet lab in vitro and in vivo studies and continuing development of models towards that, especially that might be able to leverage gene-specific and SNP-specific cell lines. And then, of course, continuing to engage the public and community through awesome events like this. I think, especially for rare disease spaces, knowledge is everything. So for someone like me who works in the industry and has never heard of NF2, now I have a special place in my heart for this disease. So I really do encourage events like these, continue growing, really continue reaching out to not only patients to make their data available, but also for people like us to continue engaging in the work. And so with that, thank you.
That was awesome. We have time for just one question. Anybody? Anybody? Going once, going twice. Rack does a lot of stuff, so what about side effects?
We will worry about side effects once we have efficacy.
Thank you very much. Another big round of applause for this team. They were awesome. All right. Please exit stage left. Ono is going to pick the next team at random. We do not know whether that will be a remote or in-person team, so be ready.
Hot dog, not hot dog.
Hot dog, not hot dog? What a fantastic team name. From generally fun people. Generally. All right. 3, 2, 1, go.
Thank you, everybody. We are the team Edge No Edge, which means hot dog, no hot dog. And at the very end, you guys are going to see why we put that name. So I just want to say that this is a very diverse team and very much fun to work with. So I'm bioinformatics. So of course, Sofia is a developmental biology and genomics. And then Luis is a translational science protein person. And then Rod is a data science person. And Mark is a machine learning person. So next slide. Oh, thanks. Yes, of course. So the approach that we follow, we wanted to set a baseline. The baseline was set up a baseline with the standard tumor normal analysis, and then based with our methods, then be able to compare against that to see what is the accuracy of our methods. So the standard tumor normal analysis was initially was the tumor, the normal, and the sibling. We included a few other samples, but was mostly with those three. And then we sort of pulled the somatic variants from there. But the important part, we followed a regions of interest approach where we merge or intersect two set of genes, one of them that was created with co-expression analysis, and then the second one with the list of genes that were created from high impact variants in the protein. And then at the very end, we sort of followed a variant filtering approach where we did interpretation of the variants, and at the end we sort of did a few variant scoring algorithms. And then of course a big part of that was the regions of interest.
So I'm a developmental biologist, and I was really interested in looking at functional relationships of genes that are expressed at the same time and at the same space. So we use a data set where there's co-expression data and try to look for interactants with NF2 and interactants of those interactants, and try to merge those genes or the gene regions to the variants of interest. And from there we found, so we had high impact variants that intersect, so 107 high impact variants, and 4 of them intersected with the co-expressed gene list set. Some of them are enlisted above. Obviously, if we reduce the impact of the variants, we increase the number of co-expressed genes. So we stayed with these first 5 genes. And those are the genes that we were interested in. And this is kind of the network, how related they are. So here's NF2, and this is one of the genes that it's associated with, and there are other 2 over there. So,
all right, so based on our analysis, we were interested in seeing how NF2 looked like, whether there were any mutations like stop codons, stuff like that. So out of our analysis, we picked 2 proteins, NF2 and EGFR. In NF2, we actually found 2 premature stop codons. And in EGFR, we found a bunch of structural interaction variant mutations, which is basically these mutations are synonymous mutations. However, they will affect the interactions of the polypeptide structure in the protein. These synonymous mutations that we found in EGFR2 might affect integration of the protein into the membrane, which can also have deleterious effects or enhancing effects. So next slide. I'm a structural biologist by training, so having seen these the truncated mutation in NF2, we were able to look up a complex. So this is basically NF2 in complex with LATS1, which is a tumor suppressor gene. So the yellow part is Anno's version of Merlin, and the red part is the part that Anno's Merlin is missing. So this is the same complex with the part that Anno is missing. So as you can see, this truncated Merlin cannot interact with LATS1, which has a bunch of downstream effects. And this also opens the possibility as a structural biologist to look at LATS1 as a possible target, like maybe perhaps by inhibiting it or treating it in some other way.
Next. Yeah. Okay, so as you can see, the focus of our approach was to narrow down our targets for research, first with looking at co-expression data to find probable expression targets that are possibly interacting with NF2. Our next thing we wanted to do after that was to rank the genetic variants on the mutations that we could find. We were working with the 1,000 genomes, and we cut it down basically to the co-expressed genes to make a smaller data set. And then as we were trying to develop a ranking model, we ran into two problems. We didn't have a large enough data set to work off of to train the model, and the second was, in place of that, our models were basically replicating already accepted statistical models based off of domain knowledge. What we ended up coming across, though, fortunately, is in Nature we found a paper that uses a boosted random forest that's been trained specifically to look for variants and rank them. And what's unique about it is it combines domain knowledge from the established measures that they're looking to improve upon, and that gives us a base for our learning. And then what they added to it is about 298 features, but it's actually 3 separate vectors of 99 features plus 1 constant. But basically what they're looking at on there is looking at evolutionary constraints between aligned human, mammalian, and vertebrate to look for evolutionarily conserved things. And that helps to create basically an environment where machine learning can create its own knowledge on top of the existing domain knowledge. What we did with this is we used their pre-trained model, so we couldn't compete with it. But it worked well for us. We were able to combine this with our reduced data set to come up with a set of targets to basically research, and then through their research on those, we were able to pinpoint the stuff we were looking at in the earlier slides. For future work, though, we would be, with a larger data set, possibly for more NF2 patients, we could use a similar model to probably look at even more possible variant features beyond just what we're looking at here, and basically train our own model off of it.
So and then, so this is the main idea that we had out of the hot dog, no hot dog idea. So we wanted to train the model and then get the variants to say, yes, NF2, no NF2. So that was the initial idea, where we are going to build the clustering, and we're going to be able to say, based on a set of variants or a genome, we're going to be able to say NF2, no NF2. Thank you.
That was fantastic. I think we're going to move along and not take questions. Most people get very intimidated when you stand very close to them. Obviously, this team is not bothered by such things. But can we get another round of applause? They're awesome. Thank you, guys. Please exit stage left and get a picture. Picture. Wait, that way. Yeah. Awesome. All right. So Ono's going to pick the next team. The reason I have my phone in my hand is because some of the remote teams have been sending bullet points on Slack. Aha. TrueCal.
Hi, my name is Chris. I'm one part of this amazingly diverse team right here. And today we worked on variant recalibration and improving that. Why are we here today? Well, ideally we're trying to take our expand our understanding of genomic data to translate that to improving patient care. And to do that, that really relies upon good processing of information as well as interpretation. You can see that errors anywhere along this pathway will result in downstream effects. And we've chosen to focus on improving variant call pipeline outputs because it's been shown that using different methods, you can get different results. Previous work has used unsupervised learning to help improve the quality of the outputs. However, with the recent release of the Genome in a Bottle truth sets, our goal was to use these truth sets as labeled data to then use a supervised learning model to help improve the positive predictive value of the outputs. So this is the approach. We have BAM data, which passes through a variant caller into a raw VCF, then it goes into our model, which has been trained using the Genome in a Bottle data to produce a true calibrated VCF. And this is important because all of you guys have been using ONO's data, the VCF files, and so our goal was really to improve your inputs. To tell you a little bit more about the methods, here's Alex.
Okay, so I was responsible for developing the models for doing this project. So I was essentially given the true VCF, and I was kind of told, okay, figure out what to put in a data matrix for this. And I was like, okay, what do I do with this massive amount of data? So I tried to identify quality features while I was reading through the true VCF, and I identified the features which are detailed below. Each of these was explained to me in detail by Don, but I don't know very much about a lot of them. They're in the header file, though, if you're interested. There was quite a bit of missing features due to the messiness of the data, so we imputed a lot of those features using the median of the features. You could also use other metrics quite easily. And then the labels, like Chris mentioned, were compiled using the Genome in a Bottle project, and this was just kind of a Boolean that identified if it was a real variant or not, so a true-false field. So we used a very simple 3-layer feed-forward neural network in order to do this, and trained it using the Keras library in Python. This produces, in essence, a probability calculator, the probability being the result of the output layer, which is passed through the sigmoid or softmax activation in the final layer. So in essence, what we wanted to do was calculate the probability that the variant was a real variant versus a false positive, kind of like we mentioned before, and the neural network drew upon other techniques which are mentioned in the slide. As for the results, go ahead.
So unfortunately, we weren't able to pass ONO's data through this, but we did come out with a great model that we think incorporates these quality metrics of the genomic outputs. So it's on our GitHub, and we'd really love for you guys to use it, and we hope it's useful. Thank you.
That was incredibly complicated. Any questions? Okay, thank you guys. Do you mind if I send this to Justin Zook? And can he contact you? I'm absolutely serious. All right, great. Thank you very much. Please exit that way to get a picture. All right. Ono's going to pick up the next thing. You know, so there's somebody like doing video of this, and she was asking me, she said, well, you know, I don't think I'm going to film the whole thing. She said, you know, and I said, well, you know what? You should get that team because that team's amazing, and that team's amazing, and that team's amazing, and that team, and that just kept happening, and it was ridiculous. Yeah, deep drugs already went. RNA-seq cluster. RNA-seq cluster. I know you guys are here. Yeah, that's you. Yeah, come on up. This is another extremely complicated project. And it is really, really amazing what you guys and the previous team were able to do in 2 days. What's that? Oh, you're missing somebody? What? Next. Yes, you can go in 5 minutes. Okay. Here, guys, just hang out over there. You don't have to go all the way back to the back. Just there. Okay. Apparently, I have lost control of this whole situation. That's the same one.
UTC.
All right, this is definitely a remote presentation.
Okay, so skip it.
No, don't skip it. I mean, we're still going to present the remote ones, yeah.
Who's gonna do it?
I am.
Okay, great.
Yeah, I actually have notes from somebody who sent me notes very early this morning. These are also on the Hackathon channel. So Parquet plus Avro is apparently more efficient for genomic analysis on Apache Spark than native bioinformatics formats, and often smaller on disk. So that's something I'm really excited about. That's awesome. And they use the Atom command line. I don't know if you guys have looked at Atom, but it's developed by some people at Berkeley and elsewhere. And so, yeah, so this is all available. This will all be available in S3 for you guys. And so this is showing you how small the other stuff was. Here are all the methods in Atom, which is pretty cool. Like I said, this will all be available in S3. They are also working on liftover to GRCH38. Which makes me very happy. For those of you who don't know, GRCH38 has a lot of corrected errors as a reference genome, and also it has alt regions, which are often relevant. Here are some more methods, and yeah, here are some interactive genomic region joins in the Atom shell. So last, I would like to give credit to the people who actually produced this. I don't know why it's a slideshow? Oh, okay. I can't scroll down and see that. I'm going to bring up the GitHub page just so I can see the end of it. Oh, wait. Hold on. So this is actually not the people that wrote this. These are, there's two breweries that helped bring this to you. I have no idea what that means, but that's awesome. But I do know that this person is listening on Facebook. So can we please give these people, can we please give them a big round of applause? That's awesome. I'm very curious about this. Can you put the link to the breweries in the Hackathon channel? Thank you. Pro team. And then we'll go back to RNA clustering next. Wait. Pro team? Okay. Back. Yeah, we'll go back to RNA clustering. Okay. Nobody leave. Unless you've already presented. Just hold it, please. Yeah, RNA clustering, same presentation? Yeah. All right.
Is it the same presentation? Yeah. Okay.
4 minutes, folks.
All right. Hi, everyone. I'm Akshay. This is Ankita. That's Dawn, Chaitanya. We have Calvin and Paul. Today we'll be talking about predicting NF2 with neural crest data. Sorry about my voice. We had a lot of debate and arguments, and over the course of the last 2 days, I lost a lot of it. Can we go next slide? So the question we want to ask is, what are the regulatory and gene expression changes that occur in NF2 disease? And second, can we leverage these to find new drug targets? That's how we started our discussion. You know, went on for a few hours. We then realized, when we actually tried something, that there's almost no NF2 patient data that's publicly available. No transcriptomic data, no epigenetic data, very little genomic data. I mean, we have ONO's data, but N of 1 makes it hard to do any sort of bioinformatics or machine learning. So knowing that, we decided to go back to the basics. What do we know about NF2? What else can we look at to understand the regulatory networks and gene expression data to actually be able to do some bioinformatics and machine learning? Oh. All right, looks like an old version was put up. So, do you want to talk?
Yeah, so I'll just explain why we went back to the neural crest. So this slide here, we went back thinking about the basics, and the NF2, it comes from the embryonic cells of the neural crest. So shown on the left is a developing embryo, and those are migrating neural crest cells. And what happens is once the baby is born and forms these tissues, the neural crest gives rise to the normal tissues as well as neural crest tumor. So what I want you guys to take away from this is these are the types of data sets we looked for in a publicly available. So we use NCBI SRA to go back and mine for RNA-seq data from early embryonic neural crest, adult neural crest, and neural crest tumor.
Thanks. Yeah, that's exactly right. So we decided to go back to neural crest data instead of only looking at NF2, because more broadly, we would expect that neural crest data has similarities with NF2 disease. So what we did was we used the NCBI database to look at different RNA-seq data, both from NF2 mutation diseases as well as any neural crest diseases. We then pulled all the RNA-seq data from these publicly available data sets, ran it through a bioinformatics pipeline to both do the alignment and the normalization. We ran it through some ML algorithms, classics like PCA or TSNE, and then we tried to identify which genes basically accounted the most for separation between classes, such as embryonic normal neural crest cells, neural crest tumors and cancers, and other NF2 gene mutations. So in particular, we chose about 5 classes, a couple from each of these, such as meningiomas and melanomas, and tried to compare them to normal to see which genes could be used to most separate those classes, and try to identify those relationships with the goal of then being able to predict the outcomes and determine NF2 causing gene pathways and networks. So once we identified the genes, we're now at this step with things running. Our goal would then be to look at gene ontology of these top genes to determine NF2-related pathways that could then be used as potential candidate drug targets. Okay, so obviously we're having some difficulties with our slides, but in conclusion, we have a really awesome group here. It was amazing to see how diverse we were. So I'm a medical student. We have a bioinformaticist. We have a biologist. We have someone who's amazing at basically data security and optimization. We have 2 computer scientists, and with this group, we were able to basically collect 82 different data sets and put it through a pipeline in 12 hours, which is shocking to me. And yeah, I think we realized ML and genomics is something that can and definitely should happen. And with that, thank you.
That was incredible. Only a third of the teams have gone yet. I mean, the science that is coming out of this is absolutely amazing. So thank you. Guys, go take a picture. Go get a picture. That way. Yeah. Oh, no. Do you want to pull up the next one? Yeah. Ono is so excited about people's presentations that he's not pulling up the next one, but that's okay. We are doing well here. And thank you to AutoNF2. Who put the link to their slideshow right on the front of their GitHub README. That was awesome. Isn't AutoNF2 a local team? Or no? No? Okay. Hopefully I have some notes. If the person who sent me the bullet points for AutoNF2, if your name is Bill, please say something in the Hackathon channel right now. So I got notes from somebody named Bill. Anyway. What's that? They're walking in. Oh. Where did you guys go? I thought you were local. By the way, Bill, can you tell? That was amazing. You guys showed up right now. Come on up. Perfect timing. Hey guys, welcome back. It's awesome that you guys came back here. What about the rest of you guys? Come on up. Yeah. Wow. The timing is incredible. Okay, cool. Here you go.
Oh, great. Hello, everyone. I guess I can start. Neurofibromatosis type 2 is a very long known rare genetic disease, but it has an increased risk of other tumors, especially nervous system, including meningiomas. That is pretty detrimental to life. Because of it being rare and its rarity, there are limited resources that makes a challenge to identify novel therapeutic targets to further identify and understand the NF2 tumors. Additionally, understanding of the genomic aberrations or the mutations of this type of tumor is little known. Also, what makes the tumor good versus the bad or the differences is still really unknown. There are numerous clinical trials that have been performed but have failed to identify systemic medical therapies that can effectively control the relentless growth of this tumor, perhaps due to the reason of its being rare. So one of the potential solutions for such rare tumors is to find bigger data sets or bigger sample size, which is it's almost impossible to do in today's time. So what is the potential solution? Would be to be able to play with the amount of data set we have now, and that's where the deep learning component comes into existence. And my friend here would describe the whole deep learning aspect.
Sure. So we propose a two-fold approach. The first approach, we use deep learning on NF2 data to train an autoencoder. And then we interpret that model to extract biological significance. The novelty of our idea is to leverage a larger data set and then train it on our smaller available data set to extract meaningful features. So we use autoencoders, which are a type of neural network that are used to extract features from very complex data. So the autoencoder is trained to regenerate the input, and it forces the network to generate meaningful encodings of complex data. So the weights contain very important information about what features are useful. So for example, if there's a large weight from node A to node B, then that implies that node A potentially influences the biological property underlying node B in a very significant manner. So we use transfer learning because the amount of data that we had to work with initially was very small. So we train on a larger NF1 data set, and then train on our NF2 data set to get meaningful features for the NF2 disease. So how we go about doing that is we analyze the weights for the encoding, and we identify nodes in we look at nodes in the hidden layer and look at the weights that go back to the input layer, which are genes and their expression data. We identify outliers based on the weights of these nodes and pick out genes that interact interact strongly with these nodes. And these hidden nodes could potentially represent biological features such as microRNA or transcription factors.
To highlight further the biological relevance of this exciting neural net is, as you can see, there are certain genes that have been highlighted by the neural net or predicted by the neural net. These are all very well-known oncogenes, and in fact, they have also been very well studied for metastatic type of cancers, perhaps one of the major leads to really understand rare types of tumors. Also, we didn't stop there. We wanted to see how efficient is our neural net, and what we did was we took the neural net genes and performed RNA-seq differential expression analysis. And you see here is the heat map, where your left is the normal cells, or the neural crest cells, and the right is the NF2 tumors. And what we see is we could actually validate with the existing in silico technique that all these genes, such as EGFR, SOX9, AL1A, etc., were significantly upregulated in the NF2 tumor types compared to the normal, which really it was exciting for us to see, and we were very happy that this is a significant observation coming out of a neural network that was tried for the first time. And we didn't want to stop there. Because we already had the neural net working, the question we wanted to ask is, can we really use this neural net to predict other biomarkers, perhaps circulating biomarkers, so a patient walks in the hospital and tells the symptoms, and the symptoms are not very clear, and the doctor can draw blood of patients and see whether these biomarkers are specific enough for NF2 tumor type, or perhaps be able to identify or monitor the progression of the disease. And what we found was that one of the microRNAs, the microRNA 200A, this neural net could show the association between this microRNA 200A with all the predicted genes coming out of the neural net, which also, I should say, this microRNA 200A is extremely well studied and also associated with poor survival outcomes in various tumor types. So quite exciting work. For future, first of all, I should say this is the first of a kind, almost a dream come true. Because I had no idea deep learning could be applied in such a manner. And what we saw was mind-blowing based on all our observations. We could also leverage that a larger related data could effectively help us identify different targets as well as biomarkers. For example, for a smaller data set such as NF2 tumors, we could also train the neural net so that we can incorporate various other parameters to increase or better performance for the model. As I mentioned, we can identify potential therapeutic targets. Interestingly, what got me very excited to see was the EGFR in the neural net. And there are a lot of ongoing efforts and trials for EGFR inhibitor, like alnotinib in various cancer types, including NF2 tumor. So it's kind of convincing that, okay, this is a great way to proceed further to understand rare diseases such as NF2 tumors. And also it has the potential to identify circulating microRNAs for early diagnostics and perhaps just to monitor the progression of the disease. And now coming to the most important slide is the true interdisciplinary team that we could create. Myself, Joe, and all of the team members here, we are very excited and we are very happy that we were given this opportunity to really go crazy with all kinds of scientific ideas. And please feel free to come to us and email us and find us, and we would be very happy to go for further deeper scientific discussions of our findings. Thank you very much.
Wow. That's how you walk into a room and kill. So if you go out that way, Pete is going to take a picture of you and then cycle back around and come sit down, watch some of the other presentations. And I think there's a question real quick. Wait, guys.
Not a question. I hope you'll contribute this to DeepChem. I think it'd be very useful to have target tools for this. So come, yeah, let's chat about this. And in general, to everyone, if you have very cool tools you'd like to build that you'd like to get out there and get maintained, contribute it to DeepChem, come find me afterwards.
Awesome. Thanks. This is on GitHub, right? Like all of the scripts and stuff? Yes. All right. Amazing. So, paranoid androids. Is that a local team or a remote team? All right. Let's see if we can find what's going on with paranoid androids. It's an awesome team name, by the way. I guess they can hear me. So that's great. I am going to go through their slides. I don't think I have talking points for them. Why did they choose this track? They want an actionable solution. So they're looking at PTK2 expression and NF2. And they're trying a new computational approach to find I'm reading the slides too. Yeah, they're trying to intercept PTK2 and heat shock protein 90 to slow down or stop tumor growth. Okay, cool. We've seen structure of Merlin. They have I'd like to point out that they have citations that they put in their presentation, which is awesome. Wow. Yeah, so they're looking at mesothelioma, which is really terrible for a lot of people. And this is how they came to HSP90 and PTK2. They're modeling target molecules as a graph and extending deep chem. That sounds awesome. And they did a bunch of stuff, and this is really great. And no, I mean that. And they found preliminary FAK inhibitor drug candidates. So what are those drug candidates? So they did some stuff with Pymol, which is really cool. They don't put the candidates on here yet, but they should verify the efficacy of the discovered drugs. Hopefully they can hear us on Facebook. If you can hear us on Facebook, if you can post the drugs that you discovered in the next 5 seconds, that would be awesome. I'm sorry, not on Facebook, on Slack. That would be good. What's that? Oh, YouTube? Oh, they're sharing on you? We're sharing on YouTube. Oh, okay. Anyway, apparently we're live on YouTube and not Facebook. But anyway, maybe we can hear from them later. So big round of applause just in case they can't hear us on YouTube. That's awesome. All right. I'm very curious as to what those inhibitors are. Read Good. Ah, yes, the Read Good team. This is the most confident team of winning of all of the teams. Whoa. They have it together. So let's see what they have.
All right, thank you. That's a lot of confidence up front. So my name is Luke, here representing Team Read Good, joined by Lucky, Joe, and Akansh, and we are actually super impressed with everyone here. It was like we're just surrounded by a lot of talented people, and it was actually really great to see. So, disclaimer from our friends at the FDA. So we mine the internet for academic sources and general news to find additional possible treatment options. So we were doing the NLP track. So we wanted basically to see, can you mine papers and discover all the treatment options that have been suggested, whether they've been researched or just speculated for NF2. And so that's actually what we did. And of course, the purpose is to provide NF2 patients like Ano with a wide variety of treatment options. So we're looking for treatment options that can help slow or stop tumor growth rather than just deal with the side effects or affect the symptoms. So we're looking at things that were really core to the reduction in tumor growth. So our approach is, again, we started with academic journals, peer-reviewed, as well as more speculative sources, so news, anecdotal reports, other things, and we fed it into a library called OpenIE. And what OpenIE does is basically give you subject-object relation tuples from text. So it says like this causes this or this treats that. And we did a lot of custom filtering on the results that are returned through OpenIE and a lot of post-processing to try to increase the actual quality of those tuples, because there is like just about every sentence has some subject relation object tuple in it, and obviously we wanted the ones that were just treatment options or potential treatments. So going more deep into this, data sourcing, a lot of web crawling, different like meta search engines. We did a lot of data cleaning. We had to take all these PDFs and then have them be something that can actually be ingested by some of these libraries. So we had to convert all of them to plain text. We did apply those algorithms, did a lot of additional filtering and everything. We also looked at doing topic modeling with GenSim and LDA, which produces some like ridiculous sentences like that, but it does contain all the relevant words that you will want to know for a particular treatment option. And then what we did is we took the candidates and then we basically filtered them down. Dr. Marco looked at the all of the results and helped us basically determine which of these were nonsensical and which were actual more robust treatment options. Anna looked as well. Thank you for that. So the results are available. So we have a searchable database that lists all the conditions, treatments, results, and the evidence. I don't think I can click through on here, but basically you can search for them, and it has things that are more well studied. So the drug panatalib is one option. That's a treatment option. But one of the things that appeared in the more speculative sources was bee pollen. So people who are looking to explore treatments that are a lot more speculative, maybe there isn't really any hard evidence for, those showed up as well, and then we have all of those available. So, future considerations. We want additional human-in-the-loop verification. The software itself doesn't have any medical knowledge to verify efficacy and other things. We want to be able to weigh it based on maybe citations or strength of evidence. More robust filtering. We only have like really maybe a few dozen papers in here. We want to be able to source with additional data. Maybe look at a more clinically oriented natural language engine that has additional drugs and other things in there that it's able to identify. And then last but not least, we want to be able to individually tailor the treatments for particular patients and their unique cases. So depending on maybe tolerance of side effect or how advanced or the nature of the actual cancer itself can be basically considered here for future work. And so there is a mailing list that you guys can reach us at. Results are available below, and we really hope that this research can have a really big impact on patients in the future and be used for other not just NF2, but other diseases as well. Thank you.
The amazing continues. That was absolutely incredible, folks. If you'll exit stage left, Pete is going to get a picture, and we will move on to the next team. I am so glad I came here. I hope you're all glad you came here. This is great. NF2 hack. Maybe they're just going to present from the README? Yeah. Okay. NF2 hack, are you here? Going once? Going twice? I think it is remote. Yeah.
I think, can I say something about this one?
Yeah,
I mean, I talked to her on Friday night, and I'm really glad to do it. So I can say that this person has like a background in science and genomics and everything, and also was interested in art, and she gave me her business card, and we talked about it, and we talked about like generating images from the genomic data. So I think that is what she did here, like generate like beautiful images from the data. I don't know on what basis she did it, but I thought it was really interesting, and I'm actually glad that she uploaded something. Like, if you are sequencing your genome, you might as well make a beautiful picture. You can put hang on your wall.
What were those last few pictures that looked like clouds?
The cloud, yeah. Maybe it is maybe it's the NF2 gene, pretty globular, getting somewhere .png. and dots translating .jpg.
This is really very creative. If you're listening, this is amazing. And for everybody else who's watching this, check out her Patreon, because there's a lot of stuff there. And there's screenshots of the work in progress. So thank you to Kristen. I did also meet Kristen. You guys are all creative, but that was definitely very creative. Amira. Is Amira local? Oh, no, Amira is definitely not local. This was sent to me this morning. So this is a proposal, and what I can say is this is also visually very interesting. It is really, really quite neat. So, yeah, and you know, I was talking to Ono about this morning, and the thing that I find amazing about it is I'm a computational biologist. Most of us are not graphic designers. But look at what this can look like. I mean, if we could just collaborate more with graphic designers, look what we could make our science look like. I mean, imagine if you took one of the teams that presented in the last half hour or last hour and their slides looked like this. I mean, you could take over the world. So, anyway, should we? Yeah. I mean, I can. You want to talk about this? Maybe. Good. Cool. All right.
I mean, I read it. So I think the idea is that you don't only collect like genomic data from people, but you collect a lot more data and try to kind of match it up in the end. So I think they're thinking of like collecting Apple Health Kit data, or if you have a watch, collect what people eat. And you know, their lifestyle, if they do any fitness or meditation, and using that other data to maybe find other patterns and you know, be surprised of finding relationships in the future. So I think it's a really good idea to get more data than only the genomic data, and yeah, see if it matches with anything. I think that's what they did. Building bridges. Yeah, so yeah, thanks to this team.
Yeah, no, I mean, this is very pretty. By the way, there's been another slight schedule change. Ono will be presenting all the remote projects from now on, because he is better at it than I am.
I was just lucky.
Deep Chem Docs.
All right, someone improved the DeepChem documentation, so I guess that's great.
That is really nice. I mean, this is truly an altruistic project. I mean, like helping somebody with their documentation. Like, all of us should write more documentation, right? Yes, all of us should write. So that's an amazing thing. Do you want to say anything about this?
Well, thank you all so much for taking this on. I'd say the more docs we write, the easier it is to do open source drug discovery. So I hope all of you will contribute and write tutorials and get people out there. Wonderful.
Great. Wow, cool. All right, thank you to Joseph. Yeah. Don't worry, there's a lot more. C cubed. This team was always happy, never daunted.
Hi everyone. Our team is C cubed, or the Cancer Clustering Collaborative, and we really enjoyed working with each other and learning from each other's backgrounds. We have a variety of backgrounds here, from undergrad to PhD, academia, industry, and lots of different subject matter expertise. So our aim today comes from the fact that, as a lot of teams have said, NF2 is a really rare cancer and there's very little data to understand it or understand how to treat it. So our goal was to look at cancers that are known and sequenced and well characterized and ask, can we find a subset of these known cancers that are biologically similar to our NF2 patient here? And can we use such a subset to suggest treatments or particular pathways that might be relevant to our NF2 patient? So our approach, for our data set, we had the VCF of the mutations that are present in Ono's tumor genome. And for existing cancers, we took TCGA data sets showing the mutations found in patients from the Cancer Genome Atlas. Since we had limited time, we took only 576 brain cancers. And we had two strategies for clustering these. The first is based on mutational signatures. I'm happy to discuss in more detail, but in a limited time, point mutations can be divided into 96 categories based on the base that is mutated, what base it's mutated into, and the base before that and after that. So basically, what's the mutation and what sequence context does it happen in? And the biological processes that cause mutations, basically mutagens, damage DNA repair pathways, enzymes that go and cause damage to the genes, cause specific patterns of mutations across these 96 categories. And these are mutational signatures. So this is an example of a biological process that mutates 4 out of 96 categories. So the first parameter which we used for clustering was mutational signatures present in the cancers, which tells us what kind of processes are acting on these patients' DNA. The second was the genes that are mutated in these cancers, and specifically the GO terms, the GO terms of those mutated genes, which indicates the biological function that is carried out by mutated genes. So basically, what biological processes are damaged in these cancers. So to do this, we took our data sets, counted trinucleotide mutations in each genome, and extracted known mutational signatures using this R package. We also identified mutated genes in each cancer genome and identified the biological function Go annotation of each gene and counted the number of genes that are mutated in each Go term. And then we used dimensionality reduction, PCA, and clustered all these cancers using k-means. So I'll briefly present our results. We filtered Ono's VCF to identify 9,600 roughly somatic mutations in his tumor. These are high-quality variants with more than 15 reads supporting the mutation and are not present in his normal tissue. We identified the mutational pattern across the 96 types of point mutations, and we identified 229 protein coding mutations in 43 genes. So these are our clustering results. Based on the mutation signatures, we identified 7 subtypes of brain cancer. And Ono's tumor is this X over here, which maps to the least populated cluster. This cluster is mostly glioblastomas and is characterized by specific mutational signatures, including what is known as signature 3. This is signature 3. It's normally found in another very well-studied type of inherited cancer, which is the BRCA-positive breast cancers. And as well as other cancers, basically the signature is found in cancers that are defective in the homologous repair DNA repair pathway. And these cancers are specifically treated by a class of drugs called PARP inhibitors. So that's an example of how we can use this strategy to identify similar cancers and identify a treatment mechanism for our patient. We then went on to the functional clustering. So this is based on what biological pathways are damaged in these cancers. And on this basis, we identified 5 subtypes of brain cancer. And this was very interesting to us because Ono's tumor is a total outlier over here. And so functionally, our NF2 patient is very, very different from all known categories of brain cancer. So our future directions, we definitely want to extend this analysis, particularly because we see that NF2 is very different from known brain cancers. What if we go ahead and look at all types of cancers, breast cancer, lung cancer, everything we know, and try to find a cluster that's similar? Will it be similar to something unexpected. We would also love to do this using other data types, including gene expression and things that could give us a better functional characteristic of the pathways that are affected in these tumors. We want to deepen our analysis, and if we find, based on various parameters, if we find that our patient is similar to some known group of tumors, we want to look very carefully at what pathways are affected in those patients and how we can use those to identify specific mutations that we can target. And basically, we gave an example of how we can use this strategy to predict treatment, and if we continue to broaden and deepen this analysis, hopefully we'd end up with a list of possible targets. Thank you.
Wow, that was really impressive. Thank you guys very much. I cannot wait. Is all the code on GitHub? Yes. Awesome. I cannot wait to dig into it, and I'm sure I speak for a lot of people who would love to take a look at this. All right, thank you guys again. You guys are amazing. SnpPixel. Hey guys. Oh yeah, here, you drive. We are very, very close.
All right, so we're Team SnpPixel, and it's our honor to be here and work with everybody. You know, you're great guys, and I know everybody's excited for the celebration afterwards. So I will try to be as quick as possible. So, and this is also our great teammates, and we are, you know, lacking two here, and we also missing a beautiful lady, maybe make it less, you know, attractive. But anyways, so so yeah, the idea behind this SnpPixel is to find the right pixel in the genome. In this case, you know, we're working on those genome, and we're specifically designed a strategy to find the NF2 mutations. So. But, you know, we're trying to keep all the variation secrets to not to leak too much of his secrecy. But anyways, so we first annotated the SNPs. Then the approaches we took is we predict the effects of the variants on the genome based on the amino acid changes, and also based on the regulatory elements, which the variations are not in the coding exon, where it could be in the introgenic region, or like upstream 3', 5' UTRs that also affecting the expression of the gene. And also, we specifically designed a approach. We actually looked backwards from the very basics about how NF2 works, because it's a rare disease, and we screened the candidate variants based on inherited and sporadic mutations. Also we monitor for the variant frequency in the populations to give us more prioritize the mutations. So this is a basic idea of the how we, you know, from the VCF files we get, we look at the different samples. So this normal blood sample is actually on those normal tissue, where we assume that it carries a one inherited homozygous mutation, which could be the one of the cause of mutations of the NF2, and in tumor tissue, that homozygous mutation that's actually making the NF2 tumor cancer tissue to grow. So for normal non-NF2 population, we imagine that most people will carry a uppercase N, though, so that's a homozygous wild type. And so quick demo here. Oh, here, yeah. So we first we combined all the VCF files we combined all the samples into one VCF file where it lists all the samples and its conditions. And the specific strategy we designed for NF2 is, as I mentioned, so we took it to evaluation of the pathways, and somehow the picture is too big to load, but we can anyway go to the we link the pathway enrichment to Reactome, which gives us a confidence of all the pathways that affect the mutation. So we see here what is really surprising for us to see. So you see here, the most enriched pathway are actually the first top two, actually. They are all located in neuron systems, which is, a kind of, surprising and exciting to see. For that, so diving into the detail of this RSK activation pathway, we see it's partially related to EGFR pathway, where we found several intron variants and also some exon sporadic mutations on chromosome X. There are also Nature and Science papers indicating the link of chromosome X and cancer, because especially in the case of male, it has one copy of chromosome X can lead to the sporadic mutation on chromosome X could have a higher potential of cancer development. And we also noticed these. This is just one. Might be too small to see, but this is just one example of the mutations that we found. It's about 3%, right? Or 0.3. Yeah, less than 3% of that mutation, while Ono carries two, rare mutations. So that's, you're very unique. So yeah, but that's our one of the proposed pathways here is that we should give the focus on this pathway and potentially find some, maybe apply deep camming there to find drugs in this pathway. And also, since we're from Minnesota and Recombinetics, we're a gene editing company that build pig models using CRISPRs to model different diseases. And we also currently have a NF2 project going on for that. So, yeah, thanks.
That was amazing. Thank you guys so much. Please go for a picture. The earlier comments notwithstanding. Yeah, do you have Cosmos thing? Yeah, or I could talk about Cosmos thing.
Oh, yeah, absolutely.
Yeah. So I think everybody met Cosmo. Cosmo was a mentor and also working on things. And the thing he said to me most frequently was, I found another variant in Ono that I think is related to disease. So let me show you some of them. So he's really excited about a tool called MendelMD, which is an open source web server that runs lots of stuff. He found 30 genes with high impact variants, and he was particularly excited about a few of them. And I think that's really exciting. One of them he has stop insertions that he found in Ono. It's in several NF2 patients. So that's a pretty obvious mutation. So he built a tool called massdb.org, and he can visualize the structural location of that. What other variants connect? Back to the hit list. One of the judges proved that ANAPC1 is linked. So thank you for that, Margot. And he was very excited about this one, too, where two isoforms of DUB3 or USP17L with a stop gain. Oh, okay. Anybody know what a stop gain is? I'm not quite sure what a stop gain is. Oh, it's a gain of a stop. Okay, gotcha. Anyway, thank you, Alvaro. I'm really glad he's here. Anyway, he also found DUB3, yet another thing, and clearly these things really sort of contribute to parts of the network. Oh my gosh, there are more of these. Yeah, Kripa kinase, there's a frame shift mutation. It inhibits PAC1, which is also involved with NF2. And here's the last one. A gain of a stop. It's right next door to NF2. Oh yeah, he was telling me about this last night. It's right next door, which is really interesting, and I encourage him to think about deletions, and there are also deletions that are known. And so here you can see what happens when you have a deletion, which thankfully Ono does not. And so, yeah, 30 critical impact, and a lot of them are already known. So that's actually really cool. And that's Cosmo's presentation. So, Cosmo had to leave suddenly to do something really, really important. Do you want to say something about that?
Well, he called it the perfect storm, which is kind of interesting.
Yes. So lots of things that have little effects on a protein pathway all aggregated together. So really very interesting. I think, has anybody that wants to present not presented? We skipped you. Oh. Ono had a reason. This is going to be cool.
Hello, everyone. So as you can see, we have a girl-only team. So we have Ran, who is a statistician, Raquel, who is a computer scientist, and my name is Joanna, and I'm a computational biologist. And our team is called PET Mutation. So the overall goal of our project was to identify new potential drugs for schwannomas. So as a starting point, we used the results from this study that was published in 2002, and it happens to come from the lab that I work in currently. So in this study, this was the very first study that used microarrays for gene expression profiling of soft tissue sarcomas. And so soft tissue sarcomas are tumors that are derived from different types of connective tissues. And this study happened to include also 2 samples from patients with schwannomas. So based on this gene expression profiling study, there were over 1,700 genes that were identified to be either up or down regulated compared to those other types of tumors. So we used this list of the genes as the starting point. And later we used Ono's tumor data, tumor compared to normal, to search for potential actionable mutations in those genes that were shown before to be dysregulated in schwannomas. So we looked for variants present in exons and also regulatory regions of those genes, and we found several deleterious exonic variants. So by deleterious we mean that they may affect the protein function. We did some additional screening, like we checked for the mappability of the regions where those variants were identified to see if these are not just like technical artifacts of the sequencing. and two of those genes were shown before to be overexpressed in schwannomas, and one of those genes was downregulated. And then we searched NCBI resources to see if there are any drugs that are known to target any of those genes. And actually, there are two drugs that are known to inhibit one of those, one of the proteins. So, and what is very interesting is that those drugs were already shown to be active in this cell line that is called SHSY5Y, which is actually a model used in research for studying neuronal function and differentiation. So very similar type of cells that, I mean, exactly the same type of cells that schwannomas are derived from. So this is just a print screen from NCBI showing PubChem information summary about those drugs. Yeah. So yeah, you can see that it indicates that there are some compounds that may be active against this target, and one of those compounds have a very fancy name, Shambo 38752, but apparently it's registered to potentially treat nervous system disorders, and it may possibly be also an anti-cancer drug. And the second drug is inositol 3-phosphate, and it has been shown that it's implicated in sphingolipid pathway, so it's also associated with the function of neuronal cells. And yeah, and that's the summary of our findings. So yeah, we'd like to thank all of the organizers for putting this event together, and we were exciting to have the opportunity to participate in this hackathon. Thank you very much.
That was great, and also the shortest presentation we've had so far. Thank you. Please go get a picture. Yeah. That's you guys? Nope, that's somebody else. But you can pick them. Yeah.
Which one is it?
Barely Understanding? They're right there. Is Barely Understanding. Yeah?
Is there a team?
Yeah, they're right there. They're coming up. Yeah.
Hi, there you go. I'll show you this.
All right, 4 minutes.
Working? Cool. Hi, so we're team Barely Understanding. And so we trained a machine learning model. Well, did a couple things. One was look at various variants and features that might be used in a machine learning model to predict epitope binding. And the other was to sort of characterize potential epitopes for treatment and immunotherapy.
Thank you, Guy. My name is Wei from Minneapolis. I was working on identifying tumor-specific antigens from whole genome sequence data. So here's some background about how we can identify this kind of antigen for immunotherapy. So you can see the green, tiny green stuff, that is antigen. If this contains some tumor-specific mutation, we call this new antigen. So basically, we have tumor normal data, so which is perfect for our pipeline. So you can compare tumor normal and find some missense mutation and mapping to the transcriptome and chop to the peptide and predict the peptide interaction between peptide and the HLA molecules. So here's a pipeline we created about 1.5 years ago. So basically, as I said, is compare tumor normal data and filter out a set of missense mutation and mapping to peptide, and using net image CPAN to predict the binding affinities. So because we don't have honest HLA typing, genotyping data, so we cannot do any more for prediction, but we are using some common allele like HLA A0201, which is quite common in Caucasians. So using this HLA allele, we going through the pipeline, and here's a part of a result we from output from the pipeline, which a list of missense mutation count in each gene. So the second gene is the most interesting me, called as NBPF1. So they have a 21 missense variants in this gene, and if you compare a tumor normal set, and here's one A change to C, which tumor is A, normal is C, that encode different peptide. One's change the amino acid from F to C. So if you predict their binding affinity, you will see IC15. So the tumors are pretty high. Normals are, tumors are low, which means they have a strong bind, and the normal is pretty high. So which means the normal tissue will never present this set of peptide into cell surface, and the T cell will never educated by this set of peptide. So once the tumor cell produce this peptide presented in the cell surface, which means T cell never seen this before. They will think it's a non-cell, so they will attack this one. So this is a tumor specific target we're trying to identify. Once we come here, meet some awesome guys. So I ask a question. So this is a critical part for prediction the binding affinity, but HLA gene is highly polymorphic. So for some common allele, you have a lot of training data set to train your neural network. But for some rare allele, it's not that good. So I asked a question: can we do some improvement for these tools? So that is these 2 days we're doing such a thing. So I'll give mic to Guy. So yeah,
this is supposed to be proof of concept, but due to lots of time constraints, it's going to be of concept. Yeah, ignore the results. It does do better, the technique I'll talk about shortly, but this is high variance and whatnot going on. Long story short, we used a one-hot encoding on these binding sequences within the motif, and we took that data, and then we also tried to incorporate some sort of structural data. And so to do that, we used a technique from algebraic topology called persistent homology. And the idea there is that if you're math savvy, you're understanding the homology of an underlying manifold. That made no sense whatsoever. You're trying to look at the shape of your data and use some sort of feature discrimination based on that. So you can sort of run it through this black box, and then you'll get some sort of distance metric. And we presented that to the network as well. It's ready to go. We just need time to run it. So I'd be interested to see what happens, and I guess that'd be like a future result. Basically, the idea behind this data set, or the method, excuse me, is that you often have many different types of features that you want to look at, but in a variety of scientific fields, I work in neuroscience, you'll just threshold and you'll say, oh, this is like biologically possible, or give some reasoning behind it. Although that may not make any sense. So by integrating the sort of features across a variety of scales and then looking at a mathematical object that encodes all that information, you might actually get significant performance boosts. And that's been shown in a variety of fields, including archaeology, networks, neuroscience, cosmology, and a variety of other areas. So we're interested to see if this works. Basically, we, as far as the pipeline did, got crystal structures, which aren't great because maybe it's not the native form of the protein, but then we used BioPython to take alpha carbon positionings and then look at a distance matrix of all the values. You can then run this through the topological feature extraction and do a bottleneck distance, which is a various metric that this thing's numerically stable under, and then feed that to your network. So that's like ready to go. We're not sure what's going to happen. Be interesting to see.
Before we have that potentially better method to work, we did some standard comparison of the existing methods for predicting the binding affinity between the HLA and the peptide. So these two are the most state-of-the-art methods out there. They both use machine learning. They both use neural networks. One uses deep learning. One is use just one hidden layer. So you can see that for the predictions that we throw in there, they tend to cluster. So there's quite some uniformity within the low end, the high end. The low end means the two molecules are binding very tightly, and the high end means they're quite loosely. So what we are really interested in is this lower left-hand corner, where the two things bind pretty tightly. That's potential targets for this immunotherapy. So we basically zoom in and took this tight binding peptides, and we realized that there's a lot of overlap between these predictions. So even if our methods don't work, the platforms that we have developed, which is our first contribution here, actually allow us to propose some candidate peptides for immunotherapy. So some other contributions, we did some comparisons for the methods, and we realized that existing methods are potentially good enough for predicting some candidate strong binders. And lastly, we want to improve beyond the standard prediction methods out there.
That was fantastic. Thank you guys very much. I mean, I think that's very exciting. Go get a picture of your team. Who are we still waiting on?
Can you give a name?
All right, so we have a couple more teams. If you're watching this on YouTube and we haven't got your remote team, please message the Hackathon's channel now. But without further ado, Team Base Pair. There's going to be some cool graphs.
Kind of, maybe. Okay. Hi, thanks everyone for having us. My name is Max. This is Jordan and
Vince.
And we are going to be. Before I begin a discussion of what we did, I want to go over for some of the non-biologists in the room, what the underlying concepts of what we're trying to do. So there are these regions of DNA that you find called enhancers. These are non-coding regions. That means they don't actually turn into protein, but they are extremely important for the regulation of individual genes. So it's a lot of biologists don't really consider them to be very sexy because they're really hard to study. You don't get a protein product that you get to play with, and yeah, just for a lot of reasons. I'd like to change that and make them a sexy topic. So hopefully after this, you'll think so. So they'll be about 100, 1,500 base pairs long, and basically what they do is they act as binding sites for proteins to come along, specifically transcription factors or other things that facilitate the transcription of genes downstream or upstream. So they'll have some sort of spatial relationship to a gene, but that is hugely variable. And it's really hard, they can be really hard to find and really hard to characterize for these reasons. And they may be located upstream or downstream of your target genes. So with this in mind, what we want to do is try to predict some of the functional effects of these rare SNPs that we're identifying in these enhancer regions. So it's interesting because a single nucleotide polymorphism in this case can have really dramatic effects for how a protein is going to bind there, how a transcription factor or some other protein binding or DNA binding protein that facilitates transcription will bind. So what we did was we used 1,000 genome projects looking for common SNPs and finding every single rare SNP that was in Anna's genome. So everything that did not pop up in the 1,000 genomes project. Using these SNPs, we then ran them through this, the deep sea convolutional neural network, which is this model that's been developed by this group at Princeton for for predicting what the effect of a single SNP will be on the binding affinity of different chromatin binding proteins. Okay, so just a little bit of the results. So we were able to identify 236 ultra-rare SNPs in Anna's genome. So these were SNPs that didn't pop up anywhere in the 1,000 genome project. Of those, running through the deep sea convolutional, the deep sea model, we were able to identify 35 SNPs with a functional significance score of less than 0.01. So that's pretty good. There's a lot to go with this, though. We need to go further and make sure that we filtered everything out correctly, and it would be great. We didn't really have enough time. There was a lot of different things happening to compare against Anna's normal expression, although that may or may not have significance because it could be just an underlying SNP in his genome, may be affecting this in other ways. So there's a lot that can really be done with this, and I think it's really exciting the way that machine learning is changing the field of bioinformatics, especially being able to translate DNA into, so think of DNA as like sentences and words, being able to translate that, like find the grammar of DNA and be able to say, this is, like identify the functional segments. So in the past, like you're able to identify motifs, you identify a ta-ta box, things like that, these functional elements. But if we can use machine learning to identify larger functional elements and spatial regulation of these things, it'll be really awesome. So yeah, some of the things we need to do is really refine some of the machine learning and the deep learning algorithms to improve performance and extend utility. Okay, and that's just a overview of what we did. And that's our team.
I told you there was more. Great job, guys. That was awesome. I mean, like, really, yeah, we don't look enough at enhancers, in my opinion, as well. All right. A couple more teams. I know you guys are back there. I know. Ono knows, too.
I just don't know which one.
Wait. Pro team and
mutation 0?
Yep. Mutation 0. Mutation 0. Pro team has to wait. Mutation 0, welcome. By the way, one of their team members has the coolest GitHub name I've ever heard. I'll let you figure out who that is.
All right, so good evening, everyone. We are the scientists and engineer from the Mutation 0 team. So I'm Chai.
And I'm Will.
I'm Calvin.
I'm Ola. Hi.
All right, and we have also tapped 2 more members who join us online. First of all, today we're going to present you how can you rank the dataset mutation using the combined annotation scoring matrix and also use the G-network approach. I think by now everyone now very convinced that we live in an era that we have a lot of genomics data, not for NF2 in specific, but overall data in general. Say we have 1,000 genome, we have encode, and people have different way of how can be made use of this different data set here to predict which gene is important one, right? So people can use like conservation score, protein interaction, also maybe like functional element in the genome. But now our approach here, we try to find a way that we can integrate all the information here and try to actually make sense of the case at hand here. To be specific, we decided to use this model from this article in Nature from Jay Chong team. These two here, basically they're called the combined annotation dependent depletion. The idea is that is to combine all the information from the ensemble variance effect predictor, ENCODE project, and also the UCSC genome browser. And then from that, they use the support vector model to train on this data set and then try to predict what is the effect from missing individual variants in the genome. The beauty of this tool is that it predicts not just on the gene level, but also at the base pair level of actually every base pair in a genome. You can predict what is the effect that it could be on both SNP and indel as well. So now let's see how can we make use of this scoring metric to solve this issue at hand. In this case, if we compare the mutation on the, we compare the genotype from the tumor brain and from the blood, then we can figure out what are the somatic mutations that this tissue could have. And you can see here there are a lot of mutations here, and if we run even the pre-trained model on this dataset, this is going to take a lot of time. So instead of that, we also try to scope down to the set that is more specific to the NF2 gene. We use this tool here called the BioGrid, which can tell us what are the genes that are highly related to the NF2. So with that, we can boil down to a smaller set, and then we can also remove the known variants from the 1,000 genome and the SNP as well. Finally, we come down into a set of 1,600 variants. These are the distribution of those variants along the chromosome 2, 3, 11, 16, and 22 here. We can do actually more because we have the patient data plus sibling as well, right? So if we subtract the variants that we found on the patient from variant in the sibling, we can actually come up with a set of rough idea of what could be potential germline mutation. Now there's a caveat here because the sibling not gonna have the same set of chromosomes because you can get different chromosomes from your father and from your mother. So there's gonna be a whole lot of false positive here. So now in this case, we scope only on the variants on the NF2 and EGFR, and also subtract a lot of them from the 1000 genome and the BSNP as well. So this is what the result look like. You can see here that on the x-axis, that's the position on the NF2 gene, and these are the somatic mutation and germline mutation that we predict. On the y-axis, those are the normalized score of the expected potential to have functional effect. And you can see here that a lot of variants that cluster at the beginning of the gene here that could potentially have the effect, and also like somewhere near toward the end as well. Now for the germline here, we have to very careful on interpretation because this could be just the difference between Ono and his brother. It may not be actually the germline mutation. But in the conclusion, I would say that now I show you that we can possibly combine all these annotated information together and plus the this very unique data set here to prioritize what are the variants that could affect the NF2 patient. We actually have already run this CADD score on the whole genome, and we just get the result like half an hour ago. And I would think that if in the future if we can genotype like parents, or we can get other NF2 patients, now we can use that to reduce the false positive that you found in the germline mutation and really pinpoint to what are the overall landscape of both somatic and germline mutation, and then show which position in the gene that you actually should target on. Thank you.
Wow, that was awesome. Thank you, guys. All right. All right, I think we have at least one more team left. Yes. So pro team. We will. Can we find them? Here we go. Come on up, guys. I would say that I spent more time with this team than any other team. So they had a lot of questions. They learned a lot. And I think they came up with something very, very creative. So without further ado.
Thank you so much for helping us. And thank you again, Ono, for providing your data and putting this together, and for all the people who put it together. Our team name is Protein. Protein, it's supposed to be protein, but because you guys get it. We're trying to be clever and funny at the same time. My name is Mary Chris.
Mukund. Jay. Vanden. Ronald.
And we're an interdisciplinary team where I'm the biologist, computer science, biomed, and chemical engineer. So let's go forward. We know, based on the literature, that NF2 is located in the 22q chromosome, and it codes for Merlin protein. And the Merlin protein has, there's a difference between the closed conformation and the open conformation. I basically just focus on two different papers, review papers. This one and this one came from one paper, and the light pink one is from one paper. So we found that from what's known out there is that the Merlin protein is phosphorylated at 3 different sites. And with this paper, it says a closed Merlin conformation leads to activation of mitogen signaling pathway. Phosphorylated is inhibition. But for the other paper, it says that phosphorylation at these 2 sites, when it binds to AKT, it inhibits binding of Merlin with PIKE-L, which inhibits PI3 kinase activity. But it promotes polyubiquitination and promotes degradation and all that. So with this in mind, we have a question. well, how can we search for unique proteins from the genomic data? And what is the likelihood of these unique proteins can be phosphorylated? So I know that we have genetic data, right, from Ono's brother, which we consider as normal, and we have Ono's control normal, internal control blood, and then we have the tumor sample from Ono. And
so the method that we wanted to find the unique proteins relevant to the tumor, and so what we did was we took the genes from well, we took the genes from all 3 genomic files, and we found, and as you can see in the Venn diagrams, we found what was unique, specific to the tumor in comparison to Ono's blood and his brother's blood. And we studied those in terms of their probabilities for phosphorylation at different sites. So specifically, we take the VCF files, we ran them through the SNP-F tool and found the RS numbers. The RS numbers are reference SNP ID numbers. And we ran those through this eSearch script, which would give us the FASTA files, and using the FASTA files would give us the amino acid sequence. Using the amino acid sequence, we fed that into the this MuSite Deep program, which uses machine learning to predict what the probabilities are of a serine, a threonine, or another amino acid, a tyrosine, being phosphorylated or phosphorylated at a certain site. And so here.
So because that was our ideally this is how you would want to analyze this data. You want to individually look at the RS numbers. However, we are provided with 2 files. One file has Ono's blood, and the other file contains Ono's brother's blood and Ono's tumor VCF file. And so it's hard for us to do the other way of analyzing. So what we did was we just we obtained the RS numbers from Ono's blood, and we obtained the unique RS numbers. So we basically just compared the unique RS numbers from the from that one file that contains 2 samples. And from that, we found that there are 149. We compared the RS, we say RSX for the blood, and then the RSY is the unique tumor RS numbers. And from that, we found 149 unique RSZ. We just say that this is the one that we're really interested in, and we fed that through the machine learning program, and we found 218 proteins. So our result shows that we have, from the 149 unique RS numbers, 44 gave us no result through NCBI. And we found 105 results from NCBI. And this is the one that corresponds to 218 proteins. And for further studies here, if we set the cutoff, so this is the frequency of proteins that have likelihood of phosphorylation sites. So if we set the cutoff to 50, we find that there are 67 proteins. If we set the cutoff to, I have my cheat sheet. If we set the cutoff to 100, we have 29 proteins. And if we set the cutoff to 200, we have 7 proteins. So further studies for this is this is what we've accomplished this weekend. Further studies. So what we can do now is we can provide you this unique proteins that have high likelihood of phosphorylation sites. And this is the very essence of personalized medicine. So ideally, for normal biologists, you take from the rat samples, right, and then put it to human. But here we have something that he gave us his sample, and we deciphered that data, and we actually found 67 proteins if we set the cutoff to 50. And we can look that further and see and do like immunohistochemistry studies, confirm if they are even present in other tumor samples, existing tumor samples that we have in the lab that maybe UCLA or I know UCSF has, if they have NF2 patients that have formalin-fixed tissues or frozen tissues, they can cut that off and do imaging studies just to see whether or not these proteins are even present. So that's it.
Thank you. Wow. And a marathon presentation as well. If you guys can go get a picture taken. Is there anybody in this room that wants to present and has not yet presented? Oh, yeah, right. Yes. Hi, guys. Come on up. What's the name of your team again? T-Rex?
T-Rex? TRS.
TRS. That's they stayed with TRS. They told me they were going to change the name, but this is going to be great. Here, come on up. TRS.
So we are Tiffany, Rena, and Chu, astonishingly called Team TRS. And so collectively, we have experience in machine learning, software engineering, and biochemistry in academia and biotech. So we're in the general vicinity of doing data analysis for genomic sequences, but we have not ourselves ever done it before. So we took this as an opportunity to do that by taking on the ranking dataset mutations problem. Let's see. So our sequence analysis pipeline was to take the VCF files, feed them through the GetK tool sets, extract samples. Since we were limited on time, we decided to focus on chromosome 22. We took differences between Ono's tumor and his normal, as well as information from his sibling. We annotated them using ANNOVAR, and then focused really on the exonic regions and excluded the synonymous variants. So one question that we had going in, kind of based on what Marco's presentation was, is were Ono's variants somatic or germline? And so based on the data that we were given, we didn't see any SNPs or indels in Ono's blood or his siblings' blood samples, the normal samples. So we understand now that they actually are somatic mutations. And he does not carry any of them in his germline. And so biologically, so what we discovered were two mutations, single nuclear polymorphisms, one that affects exon 3 and one that affects exon 6. One has already been annotated in ClinVar, but the C to G was actually a novel discovery, and both of them are actually stop gain mutations.
So we also look at all the variants on chromosome 22. So we summarized total variants, exonic variants, and non-synonymous exonic variants for the 3 samples. And we see that there are more variance in the tumor sample than in blood normal or sibling normal. And to narrow down our list, we also compared between tumor versus normal, tumor versus sibling, and normal versus sibling. So we found there are 7 different non-synonymous exonic variants between tumor and normal blood, corresponding to 5 genes, and there are 93 different non-synonymous exonic variants between tumor and sibling blood, corresponding to 38 genes. So we're going to focus on the 7 variants and the 38 genes. So here are the 7 non-synonymous exonic variants that are present in tumor but are not present in blood normal. So we see here are the 2 stop gain mutations in NF2. And for the other 5 variants, the functions of those genes are not directly implicated in cancer. Here are the 38 genes that are present with variants that are present in tumor but not in his sibling. And we also did functional annotation for the 38 genes, and we found a few interesting genes. So for example, this COMP gene is associated with neurotransmitter function. And also MAPK is a well-known gene implicated in cancer. So it's an essential component in the MAPK signaling pathway. Another interesting gene we found is this MN1 gene. So this variant is a 3 base pair insertion. It's present in Arnold's tumor and blood samples, but not in his sibling sequence data. And the function of this gene is proto-oncogene in meningioma. So in conclusion, we identified 2 stop gains in the NF2 gene, and also we discovered a novel variant in MN1. So thank you very much.
I'd say that's a pretty clear result. Great job. Yeah. Cool. Yeah. Awesome. Did we miss anybody? I just want to triple check. Anybody want to present who hasn't presented? Wow, that was an enormous amount of work. What? There are a couple of people. Yes, there's a couple of teams who haven't presented, but I think if they don't want to present, we will let them not present. Are you sure? You sure? Okay, gotcha. So that's it. Wow, that was an enormous amount of information. So here's what we're going to do now. What I would like everybody to do is let the judges get a beer and then come back here. We'll have a judge discussion because not everybody puts stuff on the Google Doc, particularly me and Ono. And we'll talk about 3 teams, and we'll let everybody else go to happy hour. And then in about 10 minutes, we'll announce sort of the results of our finding. But regardless of like sort of the winners and so on and so forth, I just want to say the amount of science that was done in the last, 73, 2.5 days is, yeah, like 48 hours. It was absolutely incredible. So here's to all of you. Amazing job. And the only other thing I would ask is just let the judges through the beer line, and then we will have a meeting, and then we will announce what's going on. So thank you to all of you. You guys are amazing.